Now, let's talk about inheritance patterns. DNA can actually not be seen through a microscope in terms of a single double helix. It's too small. Our microscopes don't have that resolution. You can get a very blurry picture with the finest resolution. But what you can see are chromosomes, which occur when a cell is going to divide, and then the DNA coalesces into a visible form that you can see under the microscope. These are the chromosomes from a single human cell. They didn't actually line up and put numbers next to themselves to help you. Somebody else had to do that. Basically, they looked at the chromosomes from a cell, and then they made a picture of it, and they cut out the chromosomes and lined them up. And because of the conventions, put numbers 1 through 22, and then the X and the Y. But an important aspect of this is you have 2 copies of all of the chromosomes, except down there in the lower right, where there's the X and the Y. I'll come to that in a minute. But for everything else, and everything else, by the way, are called the autosomes. All of the chromosomes except for the sex chromosomes are called the autosomes. You have 2 copies. You got one from mom, one from dad. This individual, when you're looking at those 2 different chromosome ones, one of those came from mom and one came from dad, and they look at this level of resolution absolutely identical. But subtly, if you had the complete DNA sequence, you would see little differences between mom and dad's chromosomes. That's how it works. So, if you're thinking about inheritance, always good to keep in mind that for most genes, we have 2 copies, a maternal and a paternal copy. Now, if a particular glitch in DNA is going to cause something to happen as far as a disorder, it matters a lot whether one misspelled copy is sufficient to cause the condition, or whether both of them have to be misspelled before you'd see any consequence. So that means the difference between dominant and recessive. So let's talk about that. In a dominant condition, even one copy being misspelled is sufficient for the disorder to appear. I'll give you a disease example in a minute, but here is the classic sort of picture of a standard pedigree in that circumstance. In this case, the father is affected, and the green chromosome in my cartoon here is the one that carries the glitch in the DNA. The mother is unaffected. Both her chromosomes are normal. As this couple has children, the father could pass on randomly one of these chromosomes or the other. And as a result, the chance of a child that's born being affected with the same condition is 50-50. And it's 50-50 each time. It doesn't remember what it did the last time. So that in this particular circumstance, the first child ended up being affected, the next 2 were unaffected, the last one was affected, turned out 2 out of 4, which is 50-50, but it wouldn't have to be. It might have been 3 out of 4, it might have been 1 out of 4. It could have been any one of those outcomes. Each time the child is conceived, there's this 50-50 chance. That's how dominant inheritance works. The way we as geneticists record this kind of information is a pedigree, as what it's called, and we use a certain kind of descriptors. So for instance, males in our pedigrees are square and females are circles. And I didn't make that up, so don't blame me. But guys, sorry, you are squares. And the upper generation here is the parents, and these are the children. And this is the way the lines are drawn to indicate this is a nuclear family, 2 parents and 4 kids. The 4 kids, they're 2 girls, 2 boys. And this is a typical pattern of a dominant inheritance, just like I showed you a minute ago, except now on a standard pedigree drawing, where the mother in this case is affected. That's her right there. And by chance, their 4 kids, 2 of them have inherited that glitch from mom, so they're affected also. That would be a dominant inheritance. Now here's a real pedigree. In this case, the condition is one that gives rise to very high cholesterol levels in the affected individuals. Familial hypercholesterolemia, that's quite a mouthful. And the infected individuals here have cholesterols of 400, 450, really dramatically high. Disease which actually was, studying this disease was the way that people developed an idea that led to statins. We wouldn't have statins today if it were not for the study of relatively rare families that have this familial form of very high cholesterol. Now you can see here in this situation, in the top generation, here's the couple The father being affected, they had 4 kids. 2 of them turned out to have very high cholesterols, going down the next generation to the grandkids. Here's a case where the father's affected, and by chance, 3 out of 3 of their children inherited that. Again, it's 50-50 each time, but in this case, it turned out to be 3 out of 3. Over here, this affected individual, it was 1 child affected and 1 not. So if you saw a pedigree of that sort, with that inheritance pattern, you would think, dominant inheritance. There are a few published examples of Moebius syndrome that are inherited in a fashion that looks dominant, but very few indeed. Most of the time, it seems, Moebius syndrome doesn't tend to follow this inheritance pattern as best we understand it. Okay, what about recessive? How does that work? Well, again, in a recessive condition, for the condition to appear, both copies of that particular gene have to have the glitch. The only way that would happen is if both the mother and the father carry a gene that has that misspelling, and they both pass it on. So the typical finding would be a case where the father is a carrier, the mother is a carrier. Generally, they don't know they're carriers, because for a recessive disease, the carriers generally have no symptoms. But think about what would happen here if this couple has a child. There are various ways in which they can pass on their chromosomes. Each parent could pass on either the pink one or the green one. And the only way you will end up getting an affected child is if they both pass on the pink chromosome, because that's the one that's carrying the glitch. And so the odds here are 1 out of 4. In a recessive disease, 1 out of 4 times the child will be affected. Again, it has no memory, so you could imagine a family of this sort where it might be 2 out of 4, or even 3 out of 4, although that would be pretty unusual, or even all 4, and that would be really unusual, because that would be sort of against the expectation as far as the odds. So certainly when a geneticist sees a family where siblings are affected and the parents are not, we tend to think of recessive inheritance. But we can't always be sure of that. It's an inference, it's a guess. Here's an example in the more standard way, drawing squares and circles for males and females. Again, sort of half filling in the symbols as up here for carriers who are not going to be noticeable. They won't know they're carriers except by the fact that they've had an affected child. And again, it's 1 out of 4. And here's a real pedigree. In this case of the disorder sickle cell anemia, which is inherited in a recessive form. Again, the carriers, the half-filled-in symbols, have sickle cell trait. You can actually, in this case with a lab test, identify who the carriers are, but in general, they have no symptoms. In this case, 2 carriers have had a total of 5 kids. And in this case, by chance, 2 of the 5 are affected. The other 3 are not. One of them's a carrier, though. An affected individual who then has children, even if the person that they have mated with is unaffected, all of the kids will be carriers. That's how recessive inheritance works. But again, from a genetics perspective, you think recessive when you see affected siblings, but unaffected parents. Now let me tell you a little bit about what we've learned about DNA, and especially in just the last 10 years. And this gives me a chance to tell you a little bit about this remarkable international effort that I had the privilege of leading called the Human Genome Project. Human Genome Project, when it was first proposed in about 1985, was considered hopelessly ambitious and audacious and would probably not happen because it was just going to be too hard, because the goal was to read out those 3 billion letters of the DNA code. And in 1985, you could almost get a PhD for reading 1,000 letters. So imagine scaling that up from 1,000 to 3 billion in your lifetime. Well, it did get started, and I'm happy to say that in the 50th year, and almost to the month, of Watson and Crick's description of DNA, we succeeded in reading out those 3 billion letters, April 2003. The DNA in that case was actually derived from about 5 anonymous individuals who donated a blood sample, and then we took all the labels off because we thought it was better not to know whose DNA it was. It was basically a reference sequence. And it was quite a job to read that out, and along the way, a lot of technology had to get invented to make it possible. And these are some of the people who led that effort. I had the privilege of overseeing this group of 20 laboratories in 6 countries involving about 2,500 scientists who all agreed that this was such an important problem that they would work together, they wouldn't worry about who got the credit, and they gave the data away every 24 hours by putting it up on the internet so that anybody had a good idea about what to do with it could start right away. And that has really changed, by the way, the whole view of the appropriate way to conduct science in genetics. That really open access to the information as soon as you can possibly do so is a really good thing. And I was fortunate to be able, as part of this, to work with some of the most amazing people that you'll ever hope to meet and to borrow their brains, just like Woodrow Wilson indicated he did, which is a great way to get stuff done. I bet Vicki borrows the brains of all of you on the board and the rest of the foundation to get stuff done. We're always much better when we work as a team. So the human genome was in fact read out and placed up there for everybody to start to work with, but of course what we wanted to use this for was a foundation. It was pretty hard to do much with it in its form of just a lot of A, C, G, and T. We wanted to understand how this could be applied for uncovering answers to medical questions. Well, one of the things that's helped us a lot since the Genome Project was completed was the way in which the technology for reading out letters of the code has advanced. It has been breathtaking. The cost of sequencing a human genome, which sort of near the end of the Genome Project was running around $100 million to read out one person's genome, over the course of the next 10 years, has come down now to less than 10,000. I think there are very few examples, if any, in science where things have progressed that rapidly. This is a log scale. If you'll notice, the axis over there, the vertical axis, is in powers of 10, just indicating how quickly this cost has gone down. So it is now possible. If you want your entire genome sequenced and you've got $8,000 lying around, you could probably have that done, and then you'd have to figure out what to do with it, which is not so trivial. And the sequencing instruments that we use, which used to be the size of a large phone booth, have all been micro-sized. I'm holding one in my hand right now. That's a sequencing machine. It's about the size of a postage stamp. Amazing engineering feats have been achieved in the last few years. So it is now possible to take all of the DNA in those chromosomes and determine the sequence. And again, on my scale here, that says 3,000 megabases. Well, a megabase is 1 million base pairs, so 3,000 megabases is 3 billion letters. But it is still a real challenge if you have that in front of you to make sense out of it. So I want to tell you about a shortcut that many of us are using when we're trying to apply DNA sequencing to make sense of a medical problem. But let me first explain how that shortcut works. And this brings us back to the sort of basic dogma of molecular biology, which is DNA, RNA, and protein. It turns out that genes, which are packets of information on DNA that code for a particular protein, are interrupted multiple times in the course of their coding information. The part that codes for protein is called the exons. Here, 1, 2, 3, 4, 5 are the exons. And then there are these spacers in the middle of the gene, which are called introns. And those have to get spliced out in the RNA. So the RNA gets rid of the spacer and patches the exons together so that now it's really a message ready to be turned into protein. And again, remember, this is called transcription from DNA to RNA. Then the RNA has to be read in that protein factory to make protein, and that's called translation. And there is a code that takes the letters in DNA and RNA, those 4 letters, A, C, G, T, and figures out how to translate that into proteins, which are made up of amino acids, which have 20 different letters. And it's a very elaborate code that was figured out by an NIH investigator back in the 1960s, Marshall Nierenberg, that tells us if you have the DNA sequence, you know what the protein sequence will be. And I'll show you an example in a little bit of what that looks like.